Hi everyone, my name is Adrianne Wilson. I'm a PhD candidate at the University of Miami. I'm also an LMR CSC student. And today I'm going to be talking to you about a management strategy evaluation I conducted on lane snapper, a data limited species. So lane snapper are a warm water species. They're found from North Carolina throughout the Gulf of Mexico, down south to Brazil. They prefer structure, so they're found around coral reefs. Juveniles are found within mangroves and estuaries, and the adults are found further offshore in water as deep as 400 meters. They're popular recreational and commercial species. They are smaller than red snapper, but they taste just as good. So I'm studying this species because it's considered data limited. And so that means we don't have enough data to look at how well the fishery is doing. So NOAA has listed the fishery as experiencing overfishing, but we're not sure if the status is overfished. It is really popular among recreational fishers, and from 2010 to 2020, the total landings have increased from 190,000 pounds to over 1 million pounds. The annual catch limit was also increased this year from 301,000 pounds to a little over 1 million. And it's interesting because the length of maturity, 50% maturity, is 240 millimeters, but our minimum catch size is 203 millimeters. So that means we're catching this fish before they're reaching maturity and before they're able to reproduce. Lane snapper are also unique in that there's evidence of genetic divergence within the stock in the Gulf of Mexico. So that means that there may be two potentially genetically different stocks, and it may be important to manage them separately if they're very different. So the aim of my study is to examine the impacts of various management procedures on the lane snapper fishery over time. And it's difficult to do this experiment in the real world because of issues with control groups and replications. So I'm conducting a management strategy evaluation, which is a method that allows us to conduct mini experiments or simulations using known stock dynamics, fishing fleet characteristics, as well as existing management frameworks. And so we can get implications about species biology, as well as which management procedures work or perform better on the fishery. So this is a chart of the management procedures or the rules that I'm going to impose on my simulated fishery. Starting at the top, Matt, Lynn, Lim will set the minimum size limit at 50% maturity. The following three management procedures, Matt, Lynn, Lim, 8, 9, and 10, set the minimum size limit to 8, 9, and 10 inches. Now the final two are management procedures that I took from the NOAA CDAR report, which is a data limited report on lane snapper and a couple other data limited fish. I slope incrementally adjust the total allowable catch to maintain a constant catch per unit effort. And then I target incrementally adjust the TAC to reach a target CPU. So these are von Berlanti growth curves, and I'll just keep Marie for explaining that. Thank you. So the first one that we're looking at here was a study done in the Gulf of Mexico where samples were taken from 1991 to 1994. This second green curve is a study that was done in Bermuda from 92 to 96. And the NOAA CDAR report used biological data from this updated curve taken from raw data from the green curve. So you can see that the samples in the Gulf of Mexico have a less steep curve. So these fish aren't reaching their maximum size as fast as the samples from Bermuda. Now on the other side are data from a study that I conducted with the help of the Panama City Beach Lab in Southeast Fisheries Science Center. These samples were taken from 2015 to 2017 throughout the Gulf of Mexico. And so what's important here is this chart. So the growth parameter here for the CDAR report, you can see is smaller than the growth parameters that I got from my samples, as well as the maximum mean length is higher for the NOAA samples. So my fish are approaching their maximum size faster than the NOAA samples, but their maximum size is smaller overall. And so I also, in my study or in the operating model, I put a maximum age of 19. Those fish from the NOAA report, they did get fish that were age 19, but in my sample size, I only got fish at around 14, but I know that they could reach that age, so that's what I included in both models. Okay, so the performance metrics are how we measure how effective the management procedures are doing on the fishery. So We're going to measure probability of not overfishing, the spawning biomass relative to spawning biomass at maximum sustainable yield at 0.1, 0.5, and 100%. And then finally, the average yield relative to reference yield. Okay, so these are trade-off plots. What's most important is that the management procedures be in the top right corner. So we're looking at the probability of not overfishing, P100, P50, and P10, and then LTY is on the y-axis. So for the NOAA data using the NOAA biological parameters, iSlope CR was the one that met all of the criteria for the performance metrics, as well as the size limit, the minimum size limit of 10 inches. But for my study, there was only one management procedure that met all of the performance criteria, and that was the iSlope management procedure. So these are the numerical values for all of those management procedures. What's important here is that they need to be above 50% or 0.5 probability. So as you can see, the only two for the NOAA report or the Matlin Lim 10 and the iSlope, and then for my data, iSlope at the bottom. So iSlope CR49 met all of the performance metrics for both operating models and was considered the most robust. And I found that MSE is a method that requires fewer data points. So if you have a fishery that you're not sure about their discards or fishing effort or things like that, it offers a practical short-term solution for data-limited fisheries. And this can be important for fishing systems in less developed countries or artisanal fisheries where they may not have that much data. Also, the models are largely dependent on our data inputs. So you can see when I changed the biological factors, it changed which management procedures were effective. And so that Johnson study that was conducted in Bermuda may not be representative of the stock that we have in the Gulf of Mexico. And those samples were taken over 30 years ago in some cases. So because of the way that we're fishing, lane snapper, and we're moving so many from the environment now, the growth rates may have shifted since then. And also from those growth curves, you can see that the Gulf of Mexico fish did not grow as fast as the samples from Bermuda. So moving forward, it would be nice to increase our sample size and range and when we collect our samples. And so that's what my dissertation is about. I've collected samples from North Carolina, Florida, throughout the Gulf of Mexico, and even in Belize. And I also am looking at increasing the age range of those samples that we're using, because with growth curves, it's very important to have smaller fish, younger fish, as well as the oldest fish that we can find. So my samples were collected with trawling as well as hook and line. And I was able to get some juveniles from North Carolina and some young of the year fish. So I'm working on aging those right now. And then I'm also looking at the genetic divergence. So I mentioned that previous study where there could be two separate stocks in the Gulf of Mexico. I've collected tissue samples from all of those regions, and I'm looking at whether or not those populations are connected, if they're exchanging genetic information. And if they are, then maybe we can continue managing it as one stock. Or if they're different, then it may be important to start managing them separately. I'm also going to look at investigating size selectivity. So those two studies from the NOAA CDAR report used hook and line to catch all of their samples. And so it's likely they're missing some fish, like the smaller fish, the young of the year fish, as well as a lot of the older fish that are usually found really offshore, and it's pretty hard to get them. And so with that, I'd like to thank my funding source, LMRCSC, all of the fishermen who helped me collect my fish, as well as my otolith processing team. There's a lot of work to take out otoliths, and they smell. I'd like to thank the Panama City Beach Lab for teaching me how to age fish, as well as my committee members, Dr. Zeth Babcock, Robert Allman, David DA, Doug Crawford, and my parents, family, and friends.
We have time for questions, and I'd like to thank Adrianne for building in some time for technical issues. Yeah.
All right. I see you. What made you choose von Bertalanffy growth curves as opposed to other growth curves?
So a lot of the literature on lane snapper already use von Bertalanffy growth curves. So when I want to compare my data, it's nice to be using the same methodology. Yes.
I have two. Are these fish like a R-selected type fish?
I'm not sure.
So like they have like a relatively small size and they usually produce a lot?
Yeah, so they are smaller than red snapper. They reach their max size around 60 centimeters. So I'm not sure about how much they spawn, but that's a good question.
Cool. And have y'all thought about maybe seeing if there's any type of regime shifts occurring in the community?
That's I do want to look into that, and I hope with the genetic connectivity, I can kind of look at different things happening within the populations.
Awesome. Thank you.
Yeah. Yes.
So great presentation. So I have a two-part to it as well. So have they recognized these Atlantic snappers to be two different separate ecotypes, or they're just assuming that they're two different, like a subspecies or two different species?
Right. So the study that was done was using mitochondrial DNA, and it was the only study that was done. So the study that I'm conducting with my tissue samples is GBS. So I'm going to try to look into that in more depth. And lane snapper are also known to hybridize with yellowtail. So that could also be a factor.
Okay, you answered my two parts. So.
Oh, great. Any more questions? Yes, I saw your hand first.
Yeah, are you thinking of getting some fishery independent data versus the fishery independent data, because that's going to be triggering a lot of that? Mm-hmm. And how are you going to be accounting for the state differences in terms of their regulations for minimum quotas? Okay, so for the first part of your question, the growth curve that I had for my study was both fishery dependent and independent data. So that kind of covers that. And then your next question was? How will you account for the difference in the states within the Gulf of Mexico in terms of the regulations? Because they have different quotas depending on the size and also quantities. So within the Gulf of Mexico and the South Atlantic, the minimum size limit is the same. There's also, I believe the catch limit is the same in terms of the bag limit. So yeah, those kind of overlap in terms of their regulations. Their quotas, I have not looked into that closely. Thank you.